About 3 to 15% of people on Earth carry something in their blood that scientists are still working to fully understand. Not a disease. Not a defect. An absence. A missing antigen on the surface of red blood cells. Something invisible to the naked eye. Unfelt in daily life. Undetectable without a laboratory test. And yet its presence or absence has quietly shaped medical history. Sparked long-running questions that serious researchers have not yet resolved, and left a distribution pattern across human populations that points toward a history we are only beginning to read. You may carry this absence yourself, or you may be here simply because the question caught your attention and would not let go. Either way, the story of Rh-negative blood is one of the stranger, unresolved patterns in human blood biology. And tonight, we are going to follow it slowly, carefully, and honestly. From the first moment, researchers noticed something did not fit. Through the populations and parasites and ancient migrations that may hold part of the answer. All the way to the question that remains open at the end of every serious investigation into this subject. One note before we begin. This video explores Rh-negative blood through the lens of current scientific research and documented history. Where evidence is limited or actively debated among researchers, we will say so. We are not here to tell you what to believe. We are here to follow the evidence and to sit honestly with the places where it runs out. Now let your breathing slow. This story has been waiting a long time. We do not need to hurry. Chapter 1. The Blood That Doesn't Fit. Picture a laboratory, sometime around 1940. Not the clean, controlled environment of modern research. Something older and more crowded. Glass equipment on wooden benches. The faint chemical smell that settles into every surface over years of careful work. Two researchers, Karl Landsteiner and Alexander Wiener, moving through a series of experiments involving blood samples and a specific marker found on the surface of red blood cells. They had named this marker the Rhesus factor, after the Rhesus monkey used in early experimental work. Most of the blood samples they tested carried it, but some did not. No marker, just a quiet absence where something was expected to be. Those samples were labeled Rh-negative, not because something had gone wrong, not because the blood was damaged or incomplete, simply because it did not carry a specific antigen that the majority of human blood carried. The designation seemed straightforward at the time. It would not stay straightforward for long. The history of how the Rh blood group system was actually understood is more complicated than any simple account can capture. Working at nearly the same time, researchers Philip Levine and Rufus Stetson were investigating the causes of serious complications in certain pregnancies, and their findings pointed toward antigens related to, but distinct from, what Landsteiner and Wiener had identified. The two lines of research ran in parallel, intersected, and took years to fully reconcile. The picture that eventually emerged was not the product of a single clean discovery, but of multiple laboratories working through contradictory findings until a coherent framework slowly took shape. What that framework revealed, as it clarified over the following decades, was that the Rh blood group system was considerably more complex than early work suggested. Not one antigen, but a family of related markers. Not a simple binary, but a range of subtypes whose full diversity researchers are still mapping today. What matters for this story is the basic distinction that emerged from all of that work. Some people carry the D antigen on their red blood cells. Some do not. Those who do are Rh positive. Those who do not are Rh negative. And the geography of who falls into which category is where the first strange detail appears. Across most of East Asia, Rh negative blood is rare. In some populations, fewer than 1 person in 100 carries it. Across most of sub-Saharan Africa, the picture looks similar. These are enormous populations. Billions of people. and Rh-negative moves through them like a quiet exception rather than a pattern. Then move west. In Western Europe, the numbers shift noticeably. Somewhere between 15 and 17% of people in countries like France and Germany and the United Kingdom carry Rh-negative blood, enough to appear consistently in medical records, enough to shape how blood banks operate, enough to make researchers ask why the distribution looks so different from one part of the world to another. And then there is the Basque country. The Basque people, living in a region where the Pyrenees meet the Atlantic coast on the border of northern Spain and southwestern France, carry Rh-negative blood at rates reported to be higher than almost anywhere else on Earth. The exact figures vary depending on the study and the sample. And researchers are careful about citing any single number as definitive, but the direction is consistent across the literature. The Basque population stands out. And it stands out for reasons that connect to a much older and much stranger story about who these people are and where they came from. We will get to that story. But first, something more fundamental. Being Rh-negative is not about lacking something the body needs. People with Rh-negative blood live full, healthy lives. Their blood carries oxygen exactly as it should. Their immune systems function. Their hearts beat with the same rhythm as everyone else in the room. The absence of the Rh antigen does not create any disadvantage in the ordinary texture of daily life. What it does create is a set of very specific situations, rare but medically significant, where the difference between Rh-positive and Rh-negative blood becomes impossible to ignore. The most important of these involves pregnancy. When an Rh-negative woman carries a child who has inherited Rh-positive blood from the father, her immune system may, under specific circumstances, come to recognize the baby's blood as foreign. The consequences. In historical populations, without access to modern medicine, were real and sometimes devastating. That story deserves its own chapter. For now, sit with the geography. A missing antigen, distributed unevenly across the planet in a pattern that clusters in the west and thins toward the east. Concentrated most heavily in one specific population that stands apart from its neighbors in ways that go beyond blood type alone. That pattern may be a signal. What the signal means is a question researchers are still working through. But it begins, as so many questions in human genetics begin, with a people who stayed in one place while the world moved around them, and with a language that has no relatives anywhere on Earth. Chapter 2. The Basque Anomaly. There are places in the world where the past refused to be replaced. The Basque country is one of them. It sits in a corner of southwestern Europe that geography itself seems to have set apart. Mountains to the south and east. The Atlantic pushing in from the north and west. A landscape of green valleys rising into stone ridges. Rivers cutting down toward a coastline where the weather arrives hard off the ocean, and the light has a particular quality that people who have seen it tend to remember long after they have left. The people who have lived in this landscape for longer than most genetic records can clearly speak a language called Euskara. And Euskara is, in the technical vocabulary of linguistics, a language isolate. This means it has no demonstrated relationship to any other language family on Earth. Not to the Indo-European family that includes English and Spanish and French and Russian and hundreds of other languages spread across Europe and beyond. Not to any language spoken in Africa or Asia or the Americas. It stands entirely alone in the linguistic record, with a grammar and a vocabulary and a structure that appear to have developed in isolation from every other known human language. Linguists have been studying Euskara seriously for well over a century. The consensus has not changed. Its origins are unclear. Its relatives are unknown. The most honest answer to the question of where it came from is that we do not yet know. Hold that for a moment. A language with no known relatives. spoken by a population that also carries Rh-negative blood at rates significantly higher than surrounding peoples. Two forms of distinctiveness: linguistic and biological, pointing in the same direction, toward a population that has, for a very long time, been separate from the people around them in ways that left marks in both their culture and their cells. Why? The current best evidence comes from ancient DNA research, a field transformed over the past 15 years as techniques for extracting and sequencing genetic material from ancient skeletal remains have improved dramatically. Studies examining ancient remains from the Iberian Peninsula have found patterns consistent with a specific picture of Basque population history. The Basque people appear to descend in significant part from early European populations who were subsequently less affected by the waves of migration that reshaped the genetics of most other European communities over the past several thousand years. While surrounding populations were being transformed by new arrivals from the east, the Basque region experienced a degree of genetic continuity that preserved older patterns in ways that became visible only when researchers had the tools to look back far enough. This is the mechanism that geneticists call a founder effect, combined with genetic drift. When a population remains relatively small and relatively closed across many generations, the genetic variance present in the founding group can become more concentrated simply through the mathematics of inheritance in small numbers. If the founding population happened to carry Rh-negative blood at higher rates than surrounding groups, that higher rate would persist across generations and could intensify over time without requiring any selective advantage to explain it. Say that again, slowly. Because it matters. Rh-negative blood may be unusually common in the Basque population, not because it does something special for Basque people, specifically, but simply because the people who were there first happened to carry it. And the relative isolation of the population across many centuries meant it did not get diluted by the genetic material of later arrivals the way it did elsewhere in Europe. That is a satisfying explanation in outline. But it leaves something open. It explains why Rh-negative blood is concentrated in the Basque population. It is less clear about why it exists at all across such a wide swath of Western Europe, and why the global distribution looks the way it does. High in the West, low in the East, in a pattern that spans far more territory than any single isolated population can account for. Ancient DNA research is still developing. The available samples from key regions and key time periods are limited, and researchers are careful about drawing strong conclusions from incomplete data. The picture is becoming clearer as more samples are analyzed and more comparisons become possible. But it is not yet complete. The Basque anomaly remains genuinely interesting precisely because it sits at the intersection of multiple fields: linguistics, genetics, archaeology, population biology, and no single field has fully resolved what the others are pointing at. A language that stands alone, a blood type distribution that stands out, a population that stayed put while the world moved around them. These are facts worth holding carefully, not as evidence of something ancient and secret, not as proof of a special origin, but as a genuine scientific puzzle being actively worked on by researchers with tools that are improving every year, in a field that has already overturned several confident assumptions about where Europeans came from and when. That is what an honest mystery looks like. Now we move closer, from ancient landscapes and population charts into something that happens not across centuries of migration, but inside a single body. In the quiet weeks and months of a pregnancy, where the difference between Rh-positive and Rh-negative blood stops being a matter of geography and becomes something immediate and personal and very difficult to ignore. Chapter 3. When blood fights itself. Imagine being told that your body might not recognize your own child. Not as a metaphor, not as an emotional description of the distance that can grow between parents and children over years, as a literal biological fact, precise and specific and measurable in a laboratory. That your immune system. The same system that has protected you your entire life, that has quietly neutralized thousands of threats you never even knew were there, might look at the blood of the child growing inside you and classify it as something foreign, something to be fought. This is not a rare theoretical scenario. It is the medical reality at the heart of Rh-negative blood. And, understanding, it is essential to understanding why the question of why this blood type persists is not simply an academic puzzle. It is a question with a weight that generations of real families have carried. Most of them, without ever knowing what was happening or why. Here is the mechanism. When an Rh-negative woman becomes pregnant with a child who has inherited Rh-positive blood from the father, two different biological identities exist in close proximity for the first time. Under normal circumstances, the mother's blood and the baby's blood do not mix directly. The placenta functions as a barrier between them. But during delivery, or sometimes during the pregnancy itself through small bleeds or certain medical procedures, a small amount of the baby's Rh-positive blood can enter the mother's bloodstream. Her immune system encounters a marker it does not recognize as self. It does what immune systems are built to do. It responds. It catalogs the foreign antigen. It begins constructing antibodies designed to neutralize it. This process, called sensitization, happens quietly, without symptoms, without any immediate consequence for the mother or the child who triggered it. The first baby is usually unaffected. But if a sensitized Rh-negative mother becomes pregnant again with an Rh-positive child, the situation changes entirely. The antibodies her immune system built are still present. They are part of her immunological memory now, ready and specific and patient. When a new pregnancy begins, those antibodies do not wait for delivery. They cross the placenta. They enter the baby's bloodstream, and they begin destroying the baby's red blood cells, the cells that carry oxygen to every developing tissue and organ. The medical name for this condition is hemolytic disease of the fetus and newborn. In its severe forms, it can cause profound anemia, organ failure, and in historical populations without access to modern intervention, death. Not in every case, not inevitably, but reliably enough, across enough families, across enough generations, to have left its mark in historical records that researchers can now read retrospectively and recognize for what it was. Families where first children survived and subsequent children did not. Patterns of infant loss that the people living through them attributed to fate or illness or the terrible randomness of early life. Because the biological mechanism responsible would not be understood for centuries. The treatment that changed this picture was developed in the 1960s. It is called RA, immunoglobulin. It works by preventing sensitization from occurring in the first place. When given to an Rh-negative mother after potential exposure to Rh-positive blood, it clears the foreign cells from her system before her immune response can build lasting antibodies. The subsequent pregnancy proceeds without the immune system treating the developing child as a threat. This is one of the quiet achievements of 20th century medicine. Unglamorous, rarely discussed outside of obstetric contexts, but enormously consequential for the tens of millions of Rh-negative women who have given birth since it became available. Now sit with the underlying biology. An immune system attacking a developing child is, from an evolutionary standpoint, a serious problem. Reproductive success is one of the core mechanisms through which evolution operates. Traits that consistently reduce the likelihood of successful reproduction face real pressure over time. And here's a trait that, under specific but not uncommon circumstances, creates exactly this kind of reproductive cost. Not in every pregnancy, not inevitably, but reliably enough in populations without modern medical intervention to have affected real families across real generations. And yet Rh-negative blood persists. It persists at 15 to 17% across Western Europe. It persists at even higher reported rates in the Basque region. It has been present in human populations long enough to appear in ancient DNA samples and historical records alike. If the cost was strong enough and consistent enough, evolution had thousands of generations to reduce its frequency. The pressure was real, the mechanism was clear, and the trait remained. This is the question at the center of everything that follows. Not dramatic, not requiring exotic explanations to take seriously, just a quiet, persistent, genuinely unresolved puzzle sitting at the intersection of evolutionary biology and human population history. Why does Rh-negative blood persist? The search for an answer leads somewhere unexpected, away from obstetric wards and population charts and into the territory of parasites and of the invisible biological pressures that shaped the human species across the long arc of evolutionary time. Chapter 4. The Evolutionary Puzzle. Think about what a deletion means. Not in the emotional sense. In the purely biological sense, somewhere in the long history of certain human populations, a gene stopped functioning. The instructions for producing a specific antigen on the surface of red blood cells were lost, or silenced, or never passed on intact. And the people who carried this loss did not disappear. They survived. They had children. Their children had children. And the deletion traveled forward through time, generation after generation, into a fraction of the human population that exists today. That is what Rh-negative blood is. At the most fundamental level, a deletion, in certain populations, of the functional version of the Rhd gene, the gene that produces the D antigen whose absence defines Rh-negative status. Deletions in the human genome are not unusual as a category. The genome carries many regions where genes have been lost or inactivated over evolutionary time. But this particular deletion is unusual in a different sense. Not because deletions are rare in general, but because of where this one shows up and how consistently it clusters in specific populations across a specific corner of the world. A variant that common, in a pattern that specific, is not neutral background noise. It is a variant that has survived long enough to tell us something. If we can work out what it is saying, ancient DNA research has begun to provide one part of the answer. Over the past 15 years, the ability to extract and sequence genetic material from ancient skeletal remains has transformed what researchers can say about human population history. Studies examining ancient remains from sites across Europe have found evidence that the genetic variants associated with Rh-negative blood were present in early European populations, and that their distribution shifted as different groups migrated, mixed, and replaced one another across the continent over thousands of years. The picture that emerges, carefully and with appropriate uncertainty, is one of a trait present in the founding populations of Western Europe that became more concentrated in certain lineages as later migrations reshaped the broader genetic landscape. Groups arriving from the East tended to carry Rh-negative blood at lower frequencies. The populations that had been in Western Europe longer carried it at higher rates. And in the places where those older populations remained relatively undiluted, the frequency stayed high. This is consistent with the founder effect explanation described in the previous chapter. But founder effects explain distribution. They do not explain persistence in the face of cost. Picture a family living in Western Europe thousands of years ago. There is no word yet for what is happening to them. The mother has lost 2 children she could not explain, to a mechanism she could not name. In a world where the connection between blood type and pregnancy outcome was entirely invisible to everyone around her, she does not know what Rh-negative means. She does not know that the blood running through her veins carries a deletion that her children may inherit, that will interact with her husband's blood type in ways that cannot be seen or felt or predicted. She only knows that some of her children did not survive. Her surviving children carry the same deletion she carries. Their children will carry it too. Why did that deletion not simply disappear over the thousands of generations during which its reproductive cost was real and its cause was completely unknown? For a trait that carries a measurable reproductive disadvantage to remain in a population across thousands of generations, something else is needed. Either the cost was lower in historical populations than modern analysis suggests, or the trait was being maintained by some compensating advantage that offset the reproductive cost in ways that simple observation of hemolytic disease does not reveal. Researchers have taken both possibilities seriously. The argument that the cost was historically lower is worth examining. If the specific conditions required for sensitization were less commonly met due to patterns of family formation and birth intervals in ancient populations, the actual frequency of hemolytic disease may have been lower than theoretical models predict. Some researchers have explored this possibility and found it worth taking seriously, though the data required to test it rigorously across ancient populations is genuinely difficult to obtain. The argument for a compensating advantage leads somewhere more interesting. In evolutionary biology, the pattern researchers look for in cases like this is called balancing selection. It occurs when a trait carries both costs and benefits, and the two remain in equilibrium because the benefit in one context offsets the cost in another. The variant persists not because it makes everything better, but because the specific environments in which the population lives create conditions where the advantage is real enough to balance the disadvantage across many generations. The most documented example of this pattern in human genetics involves a different blood condition. Sickle cell trait persists at meaningful frequencies in populations with historical exposure to malaria, because individuals who carry one copy of the sickle cell variant have red blood cells that are inhospitable to the malaria parasite. In environments where malaria was prevalent, individuals with sickle cell trait survived to reproductive age at higher rates than individuals without it. The advantage was sufficient to maintain the variant in the population despite the serious costs it imposes when two copies are inherited. The logic is elegant. The evidence is solid, and it provides a template for thinking about Rh-negative blood. If there is a compensating advantage for Rh-negative status, it is likely to be found in exactly this kind of interaction between the blood type and a specific pathogen or parasite that was sufficiently prevalent in the environments where Rh-negative blood is most common to have created meaningful selection pressure over time. The candidate that has attracted the most serious research attention is not a dramatic one, not a predator, not a plague that swept through cities. A single-celled organism that most people have never heard of, that lives quietly inside a large fraction of all humans on Earth right now, and that has been present in human environments for the entirety of human evolutionary history. Its name is Toxoplasma gondii, and the story of its possible relationship with Rh-negative blood is one of the stranger threads in this entire investigation. Chapter 5. The Parasite Hypothesis. Right now, as you lie here listening, something may be living inside your brain. Not metaphorically. A single-celled organism called Toxoplasma gondii is often estimated to infect up to around 1/3 of the global human population, though figures vary considerably by region and by the methods used to measure them. Most of those people do not know it is there. It produces no obvious symptoms in healthy adults. It does not announce itself with fever or rash or any of the signals the body normally uses to flag an unwanted guest. It settles instead into what researchers describe as a chronic, latent state. Present but contained. Watched by the immune system, but never fully eliminated. A quiet, permanent resident in muscle tissue and, most significantly, in the brain. Its primary host is the domestic cat. Inside a cat, Toxoplasma reproduces and sheds infectious particles that can survive in soil for months. But the organism is not particular about its intermediate hosts. It can infect virtually any warm-blooded animal, rodents, livestock, birds, Humans become infected through contact with contaminated cat feces, through consumption of undercooked meat from infected animals, or through transmission from mother to child during pregnancy. Once inside a human host, the cysts it forms can persist for the lifetime of that host. In people with healthy immune systems, the standard medical view has long been that this chronic infection is essentially asymptomatic. The immune system contains the parasite. Life continues normally. The passenger stays quiet. That standard view has been quietly complicated over the past 3 decades by the work of one researcher in particular, Jaroslav Flegr, a professor of biology at Charles University in Prague, began studying Toxoplasma gondii in the mid-1990s after encountering research suggesting that infected rodents showed a specific and striking behavioral change. Rodents infected with Toxoplasma appeared to lose their innate aversion to the scent of cat urine. Instead of avoiding the smell that should have triggered immediate fear and flight, infected rodents were drawn toward it. They moved in the direction of the predator that would, if the encounter proceeded as biology intended, complete the parasite's reproductive life cycle. The behavior change served the parasite's interests with a precision that Flegr found difficult to dismiss. He began asking whether something similar might be measurable in humans. Over the following 2 decades, Flegr and colleagues published a series of studies examining populations of Toxoplasma-infected and uninfected individuals across a range of behavioral and physiological measures. The findings were, depending on your perspective, either genuinely interesting or quietly unsettling. Infected individuals showed statistically measurable differences in reaction times. In certain personality assessments, and in rates of specific accident types, the effect sizes were modest, not dramatic enough to be obvious in any individual case, but consistent enough across multiple studies to suggest something real rather than statistical noise. The research attracted significant skepticism from the mainstream scientific community, as unconventional findings generally do. Critics pointed to methodological limitations, to the difficulty of establishing causation rather than correlation, and to the challenge of ruling out confounding variables in population studies of this kind. Some specific findings from Flegr's group have not been consistently replicated by other research teams working with different populations and different methodologies. Flegr himself has been careful to characterize his conclusions as preliminary and to acknowledge the limitations of the evidence. The question of whether Toxoplasma gondii produces meaningful behavioral effects in healthy humans remains genuinely contested in the scientific literature. What connects this to Rh-negative blood is a specific observation from Flegr's research program. In studying the effects of Toxoplasma infection across different groups, Flegr's team observed that the measurable effects of infection appeared to differ between Rh-positive and Rh-negative individuals. In some of the outcomes they examined, Rh-negative individuals appeared less affected by latent Toxoplasma infection than Rh-positive individuals. The differences were not large. The research group was appropriately cautious about interpreting them, but the pattern was consistent enough across their studies to propose a hypothesis. Stated carefully, the hypothesis is this: Rh-negative individuals may have a different physiological response to Toxoplasma gondii infection than Rh-positive individuals. Whether this reflects some form of partial resistance, a different immune response, or some other mechanism is not established at the molecular level. Whether the difference is large enough or consistent enough to have influenced the evolutionary persistence of Rh-negative blood is a further question that the available evidence does not yet answer. Return to the sickle cell comparison. Sickle cell trait persists in malaria endemic regions because the slight abnormality it produces in red blood cells makes those cells inhospitable to a specific pathogen. The advantage is not general. It does not make every aspect of health better. It is specific, contextual, and tied to one particular infectious pressure in one particular type of environment. Researchers exploring the toxoplasma hypothesis are asking whether something analogous is occurring with Rh-negative blood. Whether a trait that carries a reproductive cost in one specific context might carry a protective benefit in another, against a parasite that has shared human environments throughout the entirety of human evolutionary history. The honest answer, as of the current evidence, is that this remains one of the more scientifically grounded hypotheses currently discussed in this area of research. The data supporting it is suggestive but not conclusive. The mechanism is not established at the molecular level. The epidemiological evidence needed to test it at the scale required to draw firm conclusions has not yet been generated. Fleger and colleagues continue to pursue this line of research. Other groups have examined related questions with mixed results. The field is active, and the question is open. What can be said is this: the toxoplasma hypothesis requires nothing exotic to take seriously. It requires only a common parasite, a genetic variant, and the same evolutionary logic that explains sickle cell trait and dozens of other human adaptations to specific environmental pressures. It is grounded in real research by a real scientist at a real institution, examined critically by the broader scientific community, and held appropriately open while that examination continues. It may prove correct. It may not. But it points towards something worth understanding regardless of how the specific hypothesis eventually resolves. The human body was shaped by pressures that are largely invisible to modern life. The parasites and pathogens that filled ancient environments left marks in human biology that researchers are still learning to read. The variants that persist unexpectedly in specific populations often turn out, when followed carefully enough, to reflect not randomness, but the accumulated logic of survival in environments that no longer exist in the form that shaped our ancestors. Rh-negative blood may be one of those marks. Not a superpower. Not a special inheritance from a non-human source. A quiet trace of the invisible world that surrounded the people who carried it first. Written into the blood of their descendants in a pattern that researchers are still, carefully and honestly, working to understand. Chapter 6. Blood, Inheritance, and the Stories People Tell. Close your eyes for a moment and think about the oldest story you know about your own family. Not a documented history, not a birth certificate or a census record. The kind of story that surfaces at a particular kind of gathering. When the evening has gone long and the conversation has moved past the practical and into territory where people reach for something older. Someone had a gift for healing. Someone survived something they should not have survived. End quote. Someone was marked from birth as different in a way the family still talks about, generations later, without quite being able to explain why it matters that they remember. Every family has a version of this story. And the fact that every family has a version of it tells us something important. Not about genetics, not about biology, about a need that runs through human social life as persistently as blood itself. The need to find meaning in inheritance. Long before anyone understood how genetic variants were transmitted from parent to child, long before chromosomes had been identified or DNA had been sequenced, human societies had developed elaborate frameworks for understanding why certain qualities ran in families. Why the children of skilled craftspeople so often became skilled craftspeople. Why certain families seem to produce healers, or leaders, or storytellers generation after generation. Why some people seem to carry something that others did not. Something visible in the way they moved through the world. In the particular quality of their attention. In the way difficulty seemed to reach them differently than it reached everyone else around them. The framework those societies used was blood. Not blood as a genetic carrier. Blood as a metaphor for the transmission of essence. The idea that what a person was, at some fundamental level, flowed from parent to child in the same substance that flowed through their veins. That character and capacity, and something like destiny, were heritable in a way that was deep and real and not simply a matter of circumstance or chance. This framework shaped how powerful families understood themselves across centuries of recorded history. The language it produced is still with us. Blue blood as a marker of aristocratic descent. Bloodlines as the framework for tracing inheritance across generations. The idea that certain families carried something in their blood that legitimized their authority, their position, their claim to a particular place in the social order. When we look at what modern genetics actually shows about the families that built the most elaborate stories around their own bloodlines, the picture is considerably more complicated than those stories suggest. The practice of marriage within restricted social circles, driven by the logic of keeping bloodlines pure and consolidating property, produced genetic consequences that were frequently the opposite of what the stories promised. Consanguinity, the technical term for marriage between close relatives, increases the likelihood that offspring will inherit two copies of the same recessive genetic variants, including harmful ones. The Habsburg dynasty is the most documented example. Imagine a portrait gallery in a cold stone palace somewhere in Central Europe. You walk slowly past the paintings. Generation after generation of rulers, their faces rendered in careful detail by the best artists of their time. And as you move through the centuries, something becomes visible in the faces themselves. A jaw that protrudes more with each generation. A lower lip that grows fuller and more pronounced as the decades pass. A physical trait accumulating across the portraits like a slow tide. The consequence of a dynasty that married within itself so consistently and so deliberately that certain genetic variants had nowhere to go but inward, doubling and concentrating across generations until they became visible on canvas. The pronounced jaw structure now associated with the Habsburg name is understood by modern geneticists as the consequence of recessive genetic variants becoming concentrated through repeated marriage between relatives. Not the mark of a superior bloodline. The biological cost of an ideology about bloodline purity that the actual mechanics of inheritance did not support. This is what happens when the story about blood meets the reality of how blood actually works. Where Rh-negative blood enters this history is through a more indirect route. Because Western European populations carry Rh-negative blood at higher rates than populations elsewhere. And because European aristocratic families were drawn from these same populations, those families carried Rh-negative blood at rates consistent with their population of origin. This is arithmetic, not destiny. It tells us nothing special about royal blood. It tells us only that people carry the genetic variants of the populations they come from. The stories built on this arithmetic. Stories about special bloodlines deliberately preserved across centuries. About the hidden significance of Rh-negative status in families that held power. These stories reflect something real about the human need for meaning in inheritance. But they do not reflect anything real about the genetics. What they reflect is a pattern that appears consistently across human cultures and across human time. When a group of people discovers that something about themselves is unusual, they reach for a story that makes the unusual thing significant. The story does not need to be accurate. It needs to be coherent. It needs to connect the unusual fact to something larger, to a framework that gives the individual a place in a narrative that extends beyond their own lifetime. Unusual blood type becomes ancient lineage. Ancient lineage becomes special destiny. Special destiny becomes the explanation for everything about oneself that has ever felt different or difficult or not quite aligned with the ordinary expectations of the world one was born into. This is a very human thing to do. It is also worth examining honestly. Because the stories people build around biological difference have not always been harmless, the history of blood as a social metaphor includes chapters that are genuinely dark, where the logic of bloodline purity was used to justify exclusion and harm against people defined as carrying the wrong kind of blood. Modern genetics does not support any version of that logic. What it shows instead is something more interesting and more democratic. All living humans share common ancestors within a time frame that is short by evolutionary standards. The genetic variants that make any particular population distinctive are, in almost every case, the products of specific historical events, founder effects, periods of isolation, adaptations to specific environments, rather than markers of inherent superiority or special destiny. Rh-negative blood is unusual in its distribution. It is worth studying for what it reveals about human history and human biology. But the unusualness is not a mark of election. It is a fact about population genetics in a specific corner of the world. The stories people tell about it reveal something true about the enduring human need for meaning, for connection, for the sense that the particulars of one's own biology are part of a larger story worth knowing. That need is real. And it is old. It did not begin with genetics or royal families or any of the specific stories this chapter has examined. It began the way most human things begin. In the dark. When someone looked at what made them different from their neighbors and reached for the most powerful tool available to make sense of it. A story. The question worth carrying forward is not whether the stories are beautiful. Some of them are. The question is whether they are honest. And that distinction matters more than most of us. In the moment of hearing a compelling story about who we might be, I find it entirely comfortable to admit. Chapter 7. The Conspiracy Layer, and Why It Exists. There is a version of the Rh-negative story that begins not in a laboratory, but somewhere considerably further out. It involves beings from somewhere other than Earth. It involves bloodlines engineered outside the normal boundaries of human evolution, preserved across millennia by groups who understood their significance and ensured that people carrying this blood ended up in positions of influence across centuries of recorded history. It involves the Nephilim, figures mentioned briefly in ancient religious texts, reimagined as hybrid beings whose genetic legacy persists quietly in the blood of their modern descendants. It involves a secret so large and so old that the absence of physical evidence for it is taken, by those who believe it, as proof of how completely it has been concealed. These stories exist in enormous quantity. They circulate widely. They attract genuine believers, people who have read about the unusual distribution of Rh-negative blood, who have noticed the real gap in the mainstream scientific explanation, who have perhaps experienced something about their own Rh-negative status that feels personally significant, and who have found in these narratives a framework that seems to make sense of everything at once. Dismissing those people is easy. Understanding why the stories are compelling is more useful. Start with the real gap they are filling. Science has not produced a complete explanation for why Rh-negative blood persists at the frequencies it does. The honest summary from the research community involves multiple partial explanations. Founder effects, genetic drift, possible balancing selection through mechanisms not yet confirmed, and a set of open questions that remain genuinely unresolved. This is the normal condition of scientific knowledge on complex questions. It is not a failure. It is not evidence of concealment. It is what incomplete understanding looks like when it is communicated without pretending to be more certain than it is. But incomplete understanding, communicated honestly, does not satisfy the part of the human mind that needs a complete story. The conspiracy narratives about Rh-negative blood are complete. They explain everything at once. The unusual distribution, the unresolved science, the feeling that some Rh-negative individuals describe of being somehow different, of not quite fitting the standard templates of human experience, the historical patterns of certain families carrying unusual concentrations of the blood type, all of it connects, all of it fits. The story closes in a way that science, at its current frontier, cannot close. This is the appeal of conspiracy thinking more broadly, and it deserves to be named clearly rather than talked down to. The human mind is a pattern recognition system of extraordinary power. It was built across evolutionary time to find connections, construct narratives, and turn incomplete information into complete stories. Because complete stories are what allow action and decision in a world that never provides perfect information. A mind that required certainty before acting would not have survived the environments that shaped the human species. When a real phenomenon meets a real gap in the official explanation, the pattern recognition system does what it was built to do. It fills the gap. And the filling that feels most significant, most connected to the deepest questions about human origin and destiny, tends to win in the competition for belief. Stories about extraterrestrial origin win over stories about founder effects, not because the evidence supports them, but because they feel larger. They elevate a biological fact into something cosmic. They give the person carrying Rh-negative blood a story that makes their blood type not just unusual but extraordinary. Not just statistically rare, but historically significant. Not just a genetic variant, but a connection to something ancient and powerful and not entirely of this world. That is a compelling story to carry through a life. The problem is not that it is compelling. The problem is that none of its specific claims are supported by the evidence. The claim that Rh-negative blood is genetically distinct in ways that suggest non-human origin is not supported by genetic analysis. The RhD deletion that produces Rh-negative status is a human genetic variant, documented extensively, with its molecular structure and inheritance patterns understood in considerable detail. Modern genomic tools are sensitive enough to detect genetic contributions from ancient human relatives like Neanderthals and Denisovans. Contributions that show up clearly in the DNA of people alive today. Those same tools find nothing in Rh-negative blood that falls outside the range of normal human genetic variation. Nothing that points toward a source beyond the human lineage. The claim that Rh-negative individuals are physiologically different in ways that go beyond the presence or absence of a single antigen is tested by the same immunological and behavioral research discussed in previous chapters. The findings in this area are modest, contested, and consistent with the kind of normal variation within the human species that researchers find everywhere they look carefully enough. They do not suggest a separate biological origin. The claim that ancient texts describe beings whose genetic legacy persists in modern Rh-negative populations requires interpretive leaps that the texts themselves do not support when read in their original context. The passage most commonly cited, a brief and obscure reference in an ancient religious text, has been the subject of serious scholarly debate for centuries. Connecting it to a specific modern blood type requires a chain of inference so long and so dependent on assumptions made outside the text that it tells us considerably more about the framework of the interpreter than about the meaning of the passage. The claim that organized groups have managed Rh-negative bloodlines across centuries without leaving documentary evidence runs into the most fundamental problem of large-scale long-term secrecy. Secrets at that scale, sustained across that kind of time, across that many people, in that many different social and political contexts, do not hold. The historical record is full of secrets that were supposed to be permanent and were not. There is no evidence that beings from elsewhere place this blood type in the human population. None of this means the people drawn to these narratives are foolish. It means they encountered a real phenomenon, a real gap, and a real set of stories that offered to close the gap with something large and meaningful. The response is deeply human. The need it reflects is genuine. The more useful response to these narratives is not mockery. It is to take seriously what genuine uncertainty they are responding to, and to address that uncertainty with the same honesty it deserves. The genuine uncertainty here is real. Science does not fully understand why Rh-negative blood persists at the rates it does in the populations it does. The research is active. The tools are improving. The picture is becoming clearer with each passing year, even if it is not yet complete. And honest uncertainty, held carefully rather than filled prematurely, is more interesting than any closed narrative, because it points toward real research and real questions and real discoveries that are still being made by real people in laboratories and archives around the world. The true story of how Rh-negative blood arrived in the human population and why it stayed, and what it may be quietly telling us about the long, complicated history of human evolution, is stranger and more beautiful than any invented origin could be. It just requires patience. The kind of patience that sits with an open question long enough to let the real answer come into focus. Rather than reaching for the nearest story that makes the question feel closed, that is harder. It is also more honest. And in the end, honesty about what we know and what we do not know is the only foundation that actually holds. Whether you are doing science, writing history, or simply trying to understand what it means to carry something unusual in your blood that the most careful researchers in the world are still working to fully explain. Chapter 8. What research is actually saying now. Most scientific progress arrives quietly. Not as a headline. Not as a moment of sudden clarity that reorganizes everything that came before it. Just the steady accumulation of better data, better tools, better questions, each one moving the edge of what is known a small distance, further into what was previously uncertain. This is where Rh-negative blood research sits right now. Not at a turning point. Not at the edge of a revelation. In the middle of a sustained effort across multiple fields that is producing real findings, revising old assumptions, and opening new questions that the previous generation of researchers did not have the tools to ask. To understand what that research is actually saying, it helps to separate three distinct areas of inquiry. They are often collapsed together in popular accounts, but are asking quite different questions, using quite different methods, and carrying quite different levels of certainty in their current answers. The first area is molecular genetics. Think of the Rh blood group system the way you might think of a language that turns out to have far more words than you initially realized. Most people encounter it as a simple binary, positive or negative, present or absent. But the system itself, when examined closely, reveals a considerably richer vocabulary. Multiple antigens beyond the D antigen, a second gene, called RHCE, that produces additional markers with their own distributions and their own clinical implications. A range of subtypes that differ across populations in ways that matter for transfusion medicine and organ transplantation. Research in this area over the past decade has had practical consequences, matching donors and recipients at the level of full Rh phenotype, rather than just the basic positive or negative designation, reduces the risk of transfusion reactions in patients who require multiple transfusions over time. The genomic work has also added detail to the population genetics picture, providing data consistent with the founder effect and genetic drift hypotheses described in earlier chapters, without resolving the evolutionary question definitively. The questions that molecular genetics answers well are questions about mechanism and distribution. The questions about why the distribution looks the way it does require a different kind of evidence. The second area is immunology. Some research groups have examined whether Rh blood group status correlates with differences in immune response beyond the specific mechanism of hemolytic disease. Some studies have found associations between Rh status and patterns of susceptibility or resistance to certain conditions. Others, examining different populations with different methodologies, have not found the same associations. The inconsistency across studies is itself informative. It suggests that if there is a real immune effect associated with Rh blood group status, it is likely modest in magnitude, context-dependent, and influenced by interactions with other genetic variants that differ across populations. This is consistent with what researchers generally find when examining the biological effects of common genetic variants. The effects tend to be real but small, contingent rather than absolute, and understandable only in the context of the full genetic and environmental background in which they occur. The toxoplasma research fits within this broader immunological inquiry, Fleger and colleagues at Charles University in Prague have continued publishing work on the correlates of toxoplasma infection across groups defined by Rh status. As of the most recent published work available at the time this video was made, the hypothesis that Rh-negative individuals respond differently to toxoplasma infection remains supported by some findings and contested by others. The field has not converged on a consensus, and researchers in this area are appropriately cautious about their conclusions. One specific direction that has attracted attention involves the possible role of Rh proteins in tissues beyond the blood. Rh proteins are expressed not only on red blood cells, but in other tissues, including brain tissue. Some researchers have proposed that their function in the nervous system may be relevant to understanding the behavioral correlates observed in studies of toxoplasma infection. This remains preliminary, but it represents the kind of mechanistic hypothesis that, if developed further, could either support or refute the behavioral claims more definitively than population studies alone can do. The third area is ancient DNA. Imagine a researcher sitting in a laboratory, working with a fragment of bone recovered from a burial site somewhere in Central Europe. The bone is thousands of years old. Under ordinary circumstances, the genetic material inside it would have degraded beyond recovery long ago. But under the right conditions of temperature and humidity, fragments of ancient DNA survive. And the techniques for extracting and reading those fragments have improved dramatically over the past 15 years. Within the broader program of ancient DNA research, the population genetics of blood group systems, including the Rh system, have been examined as part of larger studies of ancient human variation. The findings relevant to Rh-negative blood are consistent with the picture described in earlier chapters. Rh-negative status appears to have been present in early European populations, and its current distribution reflects the sequence of migrations and admixture events that shaped European genetics over the past several thousand years. Ancient DNA research is limited by the availability of well-preserved samples from key geographic locations and time periods. Significant gaps remain. What all three research areas share is a relationship to uncertainty that is worth naming clearly. None of them have produced the definitive answer to the central evolutionary question. The molecular genetics research clarifies mechanism and distribution, but does not explain persistence. The immunological research suggests possible functional differences, but has not confirmed a compensating advantage at the scale required to explain the evolutionary picture. The ancient DNA research is filling in the historical timeline, but has not yet produced the complete picture that would allow confident conclusions. This is what active science looks like from the inside. Not a wall of settled facts, but a moving frontier of better questions, partial answers, and the gradual replacement of confident assumptions with more carefully held uncertainty. For people who want a clean and satisfying answer about why their blood type is unusual, this situation can feel frustrating. But the alternative, accepting a complete answer that is not supported by the evidence, does not actually satisfy the underlying need. It creates the feeling of resolution while leaving the real question unanswered beneath it. The real answer is still being found. Carefully, slowly, honestly, by researchers who understand that the question is worth getting right rather than getting quick. Chapter 9. The People Who Carry It. Step back from the evolutionary timescales for a moment. Step back from the ancient DNA and the parasite hypotheses and the carefully hedged language of researchers working at the edge of what is currently known. Think about a woman sitting in a waiting room. She is somewhere in her second trimester. The chair is slightly uncomfortable. There is a form on the clipboard in her lap that asks, among other things, about her blood type. She already knows the answer. She has known it since her first prenatal appointment, when her provider explained what Rh-negative means in the context of pregnancy and what the standard protocol involves. She is not afraid exactly, but she is paying a particular kind of attention that other women in the same waiting room may not be paying. The kind of attention that comes from knowing your body carries a variable that requires management. That the pregnancy proceeding normally depends in part on a medication being administered correctly and on time. This is what Rh-negative blood means in an actual human life. In the most immediate and present tense. Right now, somewhere around 15% of the population of the United States carries Rh-negative blood. That is tens of millions of Americans. Most of them aware of their blood type, only in the way that most people are aware of facts about their own biology that rarely require active attention. For most of them, Rh-negative blood is a quiet background fact. It appears on a medical record. It becomes relevant when surgery or transfusion is needed. It is noted, accommodated, and recedes back into the background of a life shaped by 1,000 more immediately pressing things. But for a specific subset of Rh-negative individuals, the blood type becomes something considerably more present. For an Rh-negative woman carrying her first child with an Rh-positive partner, the standard of care in countries with modern prenatal medicine involves screening, monitoring, and the administration of Rh immunoglobulin at specific points during pregnancy and after delivery. This protocol is well established and highly effective at preventing the sensitization that would otherwise put subsequent pregnancies at risk. For women who receive this care consistently, the management of Rh incompatibility is straightforward. One item on the checklist of prenatal medicine, handled routinely by providers who have managed it many times before. But the clinical description does not capture what it feels like to carry this knowledge through a pregnancy. The awareness that your body may respond to your own child as a foreign presence. The dependence on a medical protocol that works precisely because it interrupts something your immune system would otherwise do automatically. The particular quality of attention you bring to every test result, every appointment, every quiet moment of wondering whether everything is proceeding as it should. For women in lower-income countries and in underserved communities within higher-income countries, access to consistent prenatal care, including reliable administration of Rh immunoglobulin, remains uneven. The gap between what the medicine can do and what people actually receive is a public health reality affecting real women with Rh-negative blood right now. Not in some historical period. In the present. This is one dimension of what Rh-negative blood means in actual human lives. A dimension that the more exotic narratives about ancient bloodlines tend to obscure entirely. There is another dimension worth examining. Over the past 2 decades, something interesting has emerged in the online landscape surrounding Rh-negative blood. Communities have formed where people who carry this blood type gather to share information, compare experiences, and develop collective frameworks for understanding what their blood type might mean about them. Some of these communities are medically focused, sharing practical information about prenatal care, blood donation, and the clinical realities of Rh compatibility that are genuinely useful for people navigating these situations. Others have moved toward the conspiracy and special origin narratives described in the previous chapter, creating spaces where people explore the idea that their Rh-negative status connects them to something ancient and significant. The psychological function of these communities is worth examining without condescension. Human beings need categories. They need ways of understanding themselves in relation to other people. Frameworks that organize the experience of similarity and difference into something meaningful. The discovery that you carry an unusual blood type, one with a documented history of scientific interest and a rich mythology surrounding it, offers exactly this kind of framework. It connects you to a community of people who share a specific biological fact. It gives that fact a history, a significance, a set of stories that make it feel meaningful rather than arbitrary. The community provides something real: connection, shared identity, the sense of being part of a story larger than an individual life. The question is whether the specific framework being used to meet those needs is honest about what the evidence supports and what it does not. The evidence supports the conclusion that Rh-negative blood is a real genetic variant with a real and incompletely understood evolutionary history. Real clinical implications in specific medical contexts, and a real and interesting distribution across human populations that reflects ancient patterns of migration and isolation. It does not support the conclusion that Rh-negative individuals share a common psychological profile, a set of special abilities, or a destiny that sets them apart from the broader human family in any fundamental sense. The difference between these two versions of what Rh-negative blood means is the difference between a genuine biological phenomenon and a projected identity. Both are real in their own way. One is real in the world. The other is real in the community that has built itself around a particular interpretation of the world. Understanding that distinction is not a dismissal of the communities or the people in them. It is a way of honoring what is actually there. A genuinely interesting genetic variant with a genuinely incomplete scientific story, already more interesting than most of the narratives built around it, and not needing to be anything other than what it is. Chapter 10. The Superpower Question. It is time to answer the question in the title, not with a reveal, not with a confirmation of everything the more exciting accounts have promised. Honestly, in the way that a question this specific deserves to be answered when the person asking it is willing to sit with a real answer rather than a satisfying one. The word superpower carries a particular kind of weight. It implies an ability that exceeds the normal range of human capacity, something that gives the person who carries it a meaningful and reliable advantage, something that can be recognized, named, and depended upon. Measured against that standard, the answer is no. Rh-negative blood does not confer any ability that exceeds normal human capacity. Claims about exceptional cognitive or sensory abilities in Rh-negative individuals are not supported by peer-reviewed research. They are patterns that communities of Rh-negative individuals have identified among themselves and interpreted as meaningful, but the methodology required to establish whether these patterns are real rather than the product of shared expectation and confirmation bias, has not been applied rigorously to the question. That is the honest answer to the superpower question, as it is usually framed. But there is a more careful version worth asking. Does Rh-negative blood confer any genuine biological difference that functions as an advantage in specific contexts? And here the answer becomes more interesting. Start with what is established. O-negative blood, which is also Rh-negative, is especially valuable in emergency transfusion situations. In circumstances where a patient's blood type is unknown and time does not permit testing, O-negative blood can be given with significantly reduced risk of a serious incompatibility reaction based on the major blood group antigens. Blood banks consistently seek more O-negative donors than they have, and the relative scarcity of this blood type within the donor pool is a real and ongoing challenge for emergency medicine. This is a genuine and concrete form of value, not a superpower, but a real biological fact with real consequences for real people in medical emergencies. Now move to what is suggested but not confirmed. The immunological research described in Chapter 8 has found evidence, modest and contested but real, that Rh blood group status may influence immune response in ways that go beyond the specific mechanism of hemolytic disease. Some studies have found associations between Rh-negative status and different patterns of response to certain conditions. The findings are not consistent across all studies and all populations, which means the effect, if real, is not large or universal. The toxoplasma research presents the most developed version of this hypothesis. If Rh-negative individuals do respond differently to Toxoplasma gondii infection, and the current evidence is suggestive rather than conclusive on this point, then Rh-negative blood may carry something that functions, in a specific ecological context, as a form of partial protection against a parasite that has shared human environments throughout the entirety of human evolutionary history. This would be, in evolutionary terms, a genuine compensating advantage, not a superpower, but possibly a limited biological trade-off, a specific, contextual biological difference that functions as an advantage in one dimension while carrying a cost in another, with the net evolutionary effect depending on the balance between the two across the environments and the timeframes in which human populations have lived. The sickle cell comparison remains the clearest template for understanding what this kind of trade-off actually looks like in biological terms. Sickle cell trait does not make its carriers stronger or more capable in any general sense. In environments where malaria is prevalent, it provides a real reduction in disease burden that translates into improved survival. The advantage is specific, contextual, and comes with its own costs in other circumstances. If the toxoplasma hypothesis proves correct, Rh-negative blood may represent a similar case. That is a genuinely interesting biological story. It is also a very different story from the superpower narrative. The superpower narrative is about general superiority, about a trait that elevates rather than simply differentiates, about carrying something that makes everything better rather than trading one thing for another. The honest biological story is about trade-offs. A genetic variant that persists not because it makes everything better, but because the specific advantages it carries in specific contexts have been sufficient. Across the long and complicated history of human populations, to balance the specific costs it imposes in others. Trade-offs are less glamorous than superpowers. They are also more true to how biology actually works. One more dimension worth addressing before we leave this question. Many Rh-negative individuals describe a subjective sense of difference, a feeling of not quite fitting the standard patterns of human experience, a perception that they process the world differently, respond to situations differently, feel things at a different register than the majority of people around them. These experiences are real. The people who describe them are describing genuine subjective states. What those experiences mean is a different question entirely. Subjective difference can arise from actual biological differences in how the nervous system processes information. From the psychological effects of knowing you carry an unusual trait and belonging to a community that has built a particular identity around that trait. From the ordinary variation in human temperament and personality that produces people who feel at the margins of whatever mainstream they find themselves in, regardless of their blood type. Distinguishing between these sources requires careful, controlled research that has not yet been done at the scale needed to produce confident conclusions. What can be said is that the subjective experience of difference is real, and that its existence does not, by itself, establish what is causing it. The honest answer to the superpower question, then, is not a single sentence. It is this. Not a superpower in any meaningful sense, but possibly a limited biological trade-off, specific and contextual, whose full nature researchers are still carefully working to understand. That answer is less exciting than what the title promised, but it is more interesting than a simple confirmation would be, because it points toward the real questions, the ones whose answers will tell us something true about human biology and human history, rather than something comforting about the special destiny of a particular fraction of the population. There is a kind of superpower in that, if you want one, not in the blood itself, in the willingness to follow the honest story wherever it leads, even when it arrives somewhere quieter than you hoped. Chapter 11. The question that remains. Come back to something simple. Not to a laboratory or a research finding or a hypothesis still waiting to be confirmed or refuted. To the fact of lying here, in the dark, in a body you did not choose, carrying biological histories you did not write. You did not select your blood type. It arrived with you, encoded in every cell, written by a process that began long before you existed, and that connects you in a way that is real and not metaphorical. to the people who carried the same variant before you. Across the generations, across the migrations, across the environmental pressures and the long, slow accumulation of evolutionary time that shaped the human species into what it is today and brought you, specifically, to this moment. This is what genetics actually offers when it is understood honestly. Not a hierarchy, not a special destiny. A connection to the long, complicated, still being understood story of a species that moved across every habitable place on the planet, adapting as it went, carrying the biological signatures of every environment it passed through, every population it merged with, every pressure it survived. Rh-negative blood is one of those signatures. Its origins are not fully understood. Its evolutionary persistence is not fully explained. The most honest summary of what science currently knows is that the picture is becoming clearer, year by year, without yet being complete. Sit with that. The picture is becoming clearer without yet being complete. This is the normal condition of scientific knowledge. Not a failure, not a cover-up, not an invitation to fill the gap with a complete mythology simply because the gap exists and complete mythologies are available. Just the ordinary reality of a species that has been asking questions about itself for as long as it has had the capacity to ask questions, and that has not yet finished answering them. The questions about Rh-negative blood that remain genuinely open are worth naming one last time. Why does the evolutionary logic that explains sickle cell trait so clearly not yet have an equivalent confirmed explanation for Rh-negative blood? What would it take to either confirm or definitively rule out the toxoplasma hypothesis? What will ancient DNA research reveal as sampling becomes denser and methods improve about the specific historical events that produce the current distribution of this blood type across human populations? These are real questions being asked by real researchers. The answers are not available yet. But they are not unavailable in principle. They are the kind of questions that careful science, given enough time and enough data, is built to answer. And when those answers arrive, they will tell us something true. Not something dramatic. Not something that confirms the more exciting stories people have built around Rh-negative blood. Something better. Something that actually happened. To actual people. In an actual world that was always more interesting than any story invented to replace it. You are the current end of a very long chain. Not the destination. Just the current end. Carrying forward what was passed to you. Living in a world that the people who passed it could not have imagined. With a body shaped by a history that most of us have barely begun to understand. Let that be enough for tonight. The body that holds you has been a long time arriving here. It knows how to rest. Let it.